Thank you very much for the presentation. Hi, I'm Micael Carrier. I'm a student in Marie Tremblay's lab in the University of Victoria, and I'm going to present my project on the Fractalkine receptor deficiency combined with maternal immune activation, a novel translational model of neurodevelopmental disorder, or it is. So we know that postnatal brain development is characterized by many processes in the brain, particularly synaptic pruning, synaptogenesis over time. And we know that many of those processes are actually mediated by macroglia in the brain. Macroglia, the resident immune cell of the brain, are involved with many of these roles, particularly synapse phagocytosis, where they're going to remove unwanted synapses. They're also involved in synaptic stripping, where they're going to physically separate synapses elements. And also they're involved in the secretion of signaling molecules in the brain. So this is really nice. And we know also that the postnatal brain development can be, as many of you, the previous talk showed, is altered by many factors, which we tend to separate into categories: the genetic predisposition of the offspring, but also all the environmental factor affecting the intrauterine gestations. Many of them are included in the pollutions, early life stress, viral infections, the diet, and all of them cause neuroinflammations in the offspring. And we know that neuroinflammations actually disrupt the brain communication in the brain, because conjointly macroglia and neuron have to communicate to ensure the proper dynamic of the brain. And one of the main receptor used by macroglia and neurons are actually the fractalkine signaling, characterized by 6-3-1 on macroglia and 6-3-CL1 on neurons. So inflammation disrupts that communication and then prevents a lot of the homeostatic processes in the brain. So this is for the, based on this, we previously in the team studied the environmental risk factor by modeling viral infection using the viral mimic poly-IC that was previously shown in other talks. And we found in adults that there's some increased repetitive behavior, decreased susceptibility, and increased anxiety behavior in a sex-dependent way in those mice. And it was super interesting because in a follow-up study, we also showed that there's synaptic changes in the dorsal hippocampus, and those actually correlated with macroglial changes in the brain, particularly based on their phagocytosis. So this was super interesting. But we know that there's a second type of factors that can affect the development. It would be the genetic factors. We know that the fractalkine signaling of macroglia is actually super important to regulate their role with neurons, particularly phagocytosis, synaptic pruning, secretion of neurotrophic factors. And a decrease in the 6-3-1 receptor signaling is actually associated with a decrease in phagocytosis and a decrease in inflammations in the brain. And when it comes to neurodevelopmental disorder, mutation in the 6-3-1 receptor is actually linked in a GWAS study made in human to neurodevelopmental disorder, particularly schizophrenia and autism. So with all this together, we thought it would be super interesting to see how we can create that deficient immune system by knocking out the genes for 6-3-1 and macroglia, and see how they would react to the prenatal challenge using the poly-IC based on their behavior, the macroglia activity, and what's happening in the dorsal hippocampus like we did before. So we breeded the 6-3-1 knockout mouse, which once they were pregnant, we injected them with the poly-IC or saline at the embryonic day 9.5. And then we waited until adulthood to look at the behavior of those mice, notably in the open field and other tests, cognitive tests that we briefly did. And then at P90, we collected the tissue, first of all, to look at the transcriptomic analysis, what genes were expressed by those mice in half brain. And also we use hippocampal section to look at the density, morphology, and the lysosomal activity of macroglia in the brain. So today I'm going to just present the female result because for sake of time, the male presented a whole different story, so it will be for another time. And a lot of it was, all the imaging was done in dorsal hippocampus, which was our previous area looked at. So for the behavioral tests, with the help of Dr. Guma, we adapted the shift analysis done, and we analyzed the double hit compared to the control in the open field in the other tests. And the open field was the only one that came out significant, but it came out significant for many of the, well, pretty much all of the data we were looking at in the matrix. So in our double hit model, we saw a decrease in travel distance. In the open field, we saw an increase in freezing episode. The mouse of the double hit tend to cross lines of the open field less often, which call it with the decreased distances, showing overall decrease in locomotion and exploration of the mouse. We also saw a decrease in the entry to the center zone because anxious mouse tend to stay on the edge of the box more than in the middle, and they also spend less time in this middle of the boxes. We also measure an increase in fecal belly in the female mice. So that was super interesting, and we saw a lot of anxiety-like behavior in these mice. So it was super interesting. And then we wanted to look at the transcriptomic analysis, starting with the expression of 6-1 that was knockout. And we showed that there's actually, like, what we thought was a decrease in the 6-1 expression following the knockout, which was expected. And following that, we did a PCA analysis, and we saw that there's a nice separation between our wild-type control and the double hit of our mouse that showed significant, well, they show a different genetic signature with the PCA analysis. So that was super nice. And then we did a pathway analysis to see what were changed in these mice. And first of all, obviously, we saw a decrease in response to chemokine and inflammatory pathway in the brain that was mainly led by the decrease in 6-1 that we knocked out. So that was nice to see. And what was surprising was actually a decrease in GABA synaptic transmissions in the brain that we analyzed. That was mainly led by a decrease in GABA receptor genes, a couple of genes that encodes for GABA receptors in the brain. So that was also very nice to see. And for what the genes were, for the genes that were upregulated, we saw some of them in ADHD, there's NH3 was upregulated, which might suggest an increase in mitochondrial activity in the double hit mouse compared to control. Chemokine-like receptor 1 is expressed by monocyte and dendritic cells to be recruited in the brain. So we thought that might be a compensatory mechanism since the communication between macula and neurons is altered. They might participate to the immune reaction more than in control. And we saw the WWOX gene that is typically the downregulation of these genes is associated with a GABAergic neural death in some disorder. And we actually saw an upregulation of these genes, which was interesting. And we may think it might be a compensatory mechanism again, where following the decrease in GABA, we saw an increase in these genes compensating or trying to bring back the GABA signaling in the brain in the adult. So after this, we wanted to look at the macrocytin, and in the dorsal hippocampus, we saw an increase in nearest neighbor distance in the macula in the brain. And then using confocal macroscopy, we saw an increase in processes arborizations and a decrease in their soma area, which was super interesting. And it might suggest that macula in the brain in this condition are trying to survey the brain more by having more processes, a smaller soma, and be more ready for what's coming up. So when we go back to our question, how will mouse offspring deficient in the fractalkine receptor react to prenatal challenge to the poly IC? We saw when we took CX201 pregnant dam and we inject them with the poly-IC during gestation, and we take the offspring, the female offspring, and we look at the behavior in adulthood, we saw some anxiety-like behavior in those mice. And then with this in mind, we thought about, we have those deficits in behavior, even though they were not expressed, they were not exposed to psychological stress. So it would be super interesting in the future to see if they would be even more susceptible or resilient, like the other talk we saw today and yesterday, exposed of like how they would react to other stressors. Also, when we take those mice and we look at the brain, we can see that there's a decrease in the GABA ergic transmission, and that nicely correlates with the behavior deficit we see, even though that was really not what we were expecting, because this is not what we saw in the MIA model that we previously described in the other study. So this was super nice. And this is all for me. I want to thank my supervisor, Marie-Ève Tremblay, and my co-supervisor, Michel Jardin-Quebec, for all their mentorship, my team for all the help they are giving me, and my collaborator, Arnaud Roy, for all the help with the transcriptomic analysis. And thank you for your attention.
Thanks so much, Micael, for a super interesting talk. If someone has a question, could they use the raise hand function? That way I can identify you on the speaker panel. Out of curiosity myself, Micael, what stressor paradigms are you guys considering looking at for your kind of future directions? Is this kind of like an acute challenge or more of like a chronic style model to try and pull out a vulnerability? Or
we use in the past a lot of social defeat. So, and also a paradigm that I personally like because I think it's very social and very important for development. So I would be interested in looking at it, but we with time tend to go more with chronic unpredictable stress with IntelliCage and other type of stress of this kind to have more a control type of standard type of stress. So I think those two options would be super interesting to look at in those animals. Yeah.
Yeah, I do think that'd be super cool to see how it came out.